Hi everyone, welcome to lecture 7 of MD6028. This is the second lecture of the viruses and human disease part of the module. So this is where we're considering viruses as causative agents of human disease. So we're looking at all the different roles viruses can play in human disease. Last lecture in the first part of this little series, we looked directly at viral pathogenesis. So we looked directly at how viruses cause disease in a host, and we talked about the way different viruses cause different types of disease and why some of these differences exist. And what we're going to talk about today will be bacteriophages. Bacteriophages are really interesting in the context of human health and disease, primarily because bacteriophages, as I'm sure you know, do not actually infect human cells. So everything we talked about in the last lecture is relevant to most viruses, but for bacteriophages, there's something else going on. We talked about how most viruses, the way they cause disease is related to the cell type they infect and the type of infection that occurs. For bacteriophages, things are different because they're not infecting human cells. However, despite this, they can still cause human disease, and that's what we want to understand today. And that's why bacteriophages get their own lecture essentially, because everything is a little bit different in terms of how they cause disease. So again, that's what we're going to talk about today. First, we're going to establish some definitions. We're going to talk about what bacteriophages are. Then we're going to talk about their life cycles and some related information. And then we're going to look into the details of how bacteriophages cause disease. Because again, it is different to how other viruses typically cause disease because, and this is a crucial point, bacteriophages are not themselves capable of infecting eukaryotic cells. They are infecting bacterial cells, and as a consequence of this, they are doing things, and we'll look at these things that ultimately result in human disease. Okay, so bacteriophages, as we said, they're not one single group either on the phylogenetic system or the phenotypic system, but we can use these systems to classify them. If we use the phylogenetic classification system, we really get two big orders of bacteriophages, so two separate large groups of bacteriophages, and then a lot of other bacteriophage families that don't fit into these two groups essentially. They're, as we've said, they're different to other viruses because they infect specifically prokaryotic cells, right? They're not infecting eukaryotic cells, and the nature of their replication cycle is different as well. However, they still meet the criteria to be defined as viruses. And we've said the key criteria to be defined as a virus is the nature of living as an obligate parasite. So a virus has to infect a cell in order to replicate itself. And bacteriophages fit this definition. It's just the cell type is a specific cell type. And the other criteria is what are the biological components of a bacteriophage or a virus, and it's the same because bacteriophages are viruses, and this is why we classify them in that way. It's genetic material surrounded by protein. The only kind of exception in the case of bacteriophages is the fact that they have this long kind of body-like structure almost with these appendage-like structures on the end of that, which is what they use to attach to the surface of a cell. But this makes no matter really because they still have the key characteristics of a virus. They still have the nucleic acid and they still have proteins surrounding this. And again, that's what we see here. We have the capsid, so this is the protein shell. Sometimes we refer to it as the head of the bacteriophage because unlike other viruses, bacteriophages have this kind of directional polarity, don't they? They have a top and a bottom. And this capsid, the head, is at the top. And then there's this kind of long structure, which we could think of the body or the tail with these appendages attached. Okay, again, we can classify them using the normal classification systems we've talked about. We can think about phylogenetics, we can think about Baltimore phenotypic classification, but the important thing to remember is these are not one big phylogenetic group, and these are types of viruses. So again, the principles that apply to normal viruses apply as well. And one of those principles is that we can't classify these higher than order in the phylogenetic classification system. So just like we can't determine effectively how the existing groups of viruses that we've talked about are related to other forms of life. So all the types of viruses that we've talked about in past lectures, we've been very clear in saying that they don't fit onto the tree of life, and we can't classify them higher than order. The exact same thing is true for bacteriophages, because again, they are viruses. So we can classify many bacteriophages into two orders, which we can see on the screen here. Virals, that's the suffix meaning order. However, there's also a lot of bacteriophages that don't fit into these orders as well. So really consistent with what we understand about the other viruses, which is that the other viruses can sometimes be fit into orders, but never any higher than order. And a lot of viruses actually can't be fit into the existing orders at all. Okay, and these are some example families. So again, Viridae is the family suffix. So if you see the suffix Viridae, we're talking about a family. And these are families existing in the order Cordovirales. So again, virals is the order suffix. Okay, and again, we can look at the terminology here and we can break it down and make sure that's really clear. So virals, the order, Viridae is the family. If we look at the actual term bacteriophage, we know phage means to eat, macrophage, right? And phagocytosis, and bacteria refers to bacteria. So again, it's bacteria eater, really indicating the fact that these infect and often lyse bacterial cells. We can see Incertae sedis being written in the phylogenetic classification system. So again, this is just a shot from Wikipedia, which I think is really interesting to use because we can go on the Wikipedia articles ourselves and look at the different viruses, look at the different bacteriophages and see this for ourselves. Sometimes they'll be classified into an order, but any higher than that, there's either nothing there or there's this Incertae sedis, which essentially means unknown, so placement unknown. Because again, bacteriophages, they're not going to be classified any higher than order. Okay, so hopefully there we've established what bacteriophages are, so what we mean when we're using the term bacteriophages. And again, bacteriophages are just viruses, they're just a group of viruses that are capable of infecting bacteria. They meet all the criteria to be defined as viruses. They have the same kind of issues associated when trying to classify them phylogenetically. When we look at them physically, so what are their components, we see they are a nucleic acid surrounded by protein. And when we think about the inherent nature of their existence, we see this is completely consistent with the nature of the existence of the other viruses. And by that we mean they are obligate parasites. So they're obligated to parasitize host cells to propagate. The only real distinction is the fact that bacteriophages have evolved down a slightly different evolutionary pathway because they have evolved a specificity to infect bacterial cells rather than the eukaryotic cells that the other viruses have evolved to infect. And as a result of this slightly different evolutionary pathway, we see some other differences as well, which are structural differences. Again, just the overall shape of the virus, with it having this tail-like or body structure, some appendages attached to that. So the key similarities are there, but there are these fairly superficial differences. Okay, so what we want to do now is look at the life cycles of these bacteriophages. So again, you'll see these life cycles are very, very similar and very consistent with the life cycles of the other viruses. However, there are some distinctions there. There are some differences. So the overall purpose of the life cycle and the overall steps are not really too dissimilar. or at least the end point of each step is the same. However, there are some distinctions that we want to understand. And crucially, we want to understand that there is a distinction between the two different types of life cycle that bacteriophages can engage in. So there are two different types of bacteriophage life cycle. There's the virulent life cycle and there is the temperate life cycle, and these are really important in the context of human health and disease, so that's what we're going to talk about now. So we can define the two life cycles as being virulent or temperate, and we can also say this as virulent or lysogenic. Lysogenic basically means non-lytic, virulent means lytic. And as we know from last lecture, lytic essentially means, and from other modules as well, lytic means destroying the cell, so causing the cell to lose integrity and die and break apart. And this is a big part of how many viruses cause disease. So bacteriophages will infect bacteria, and they can have one of these two life cycles that they engage with, a lytic life cycle or a non-lytic life cycle, also known as a virulent life cycle or a temperate life cycle. Temperate life cycle can also be referred to as a lysogenic life cycle. Okay, so a lot of terms here, but the key takeaway is the life cycle can either result in the death of the cell that is being infected, and again this is a bacterial cell, or it can result in the continued existence of the cell, so it can not result in the death of the cell, so therefore not lyse the cell. Okay. And hopefully we can see this demonstrated clearly in the diagram on the right. So we have a lytic life cycle on the left hand side of this diagram, and on the right hand side we have a lysogenic life cycle. The first takeaway is that when the virus attaches to the outside of the cell, it doesn't actually fuse with the cell, so it doesn't enter into the cell in the same way that other viruses do. Bacteriophages instead essentially inject their genetic material into the cell. Again, the end point here is the same, and the end point is the nucleic acid is able to exist within the cell, so it's able to be copied and it's able to be expressed. However, how the virus is getting there is slightly different because it is injecting its genome rather than fusing with the membrane or anything like that. Obviously, that wouldn't apply to these cell types. It just couldn't happen. And neither does the virus enter whole. None of these things happen. What happens instead is the bacteriophage injects its genetic material into the bacterial cell. If we're looking at the lytic life cycle first, this phage genetic material can be expressed and replicated and all the normal things that we know happen as part of virus infection of a cell, and as a result new phage particles are produced, so new bacteriophage particles are produced, and as an end result of this process the cell is lysed, and these bacteriophage particles are released into the local environment where they can infect other cells and potentially spread and spread and spread and spread. So this is pretty consistent with what we see about the lytic viruses we've talked about so far. So some of the details are different, but essentially we have massive numbers of progeny being produced, so massive numbers of virus particles being produced, and this ultimately leads to the death of the infected cell. This death is lysis, and the new virus particles are released into the local environment. Okay, so now we can look at the lysogenic life cycle, and we can see there are some key differences. One key difference being the fact that in the lysogenic cycle, the bacterial cell that's infected does not die. It's not lysed. It is able to continue existing. It is able to continue dividing. The other key difference is the fact that what we call a prophage has been formed. So when the bacteriophage injects its genetic material into the bacterial cell, this genetic material integrates with the bacterial cell genome, and in doing so it forms what we call the prophage. Now the prophage is the bacterial genome with the bacteriophage genome integrated. So it's the bacterial genome plus the bacteriophage genome. Now what this essentially means is the bacteriophage has inserted its genome into the bacterial cell genome, and this bacterial cell can continue growing and growing and replicating. So the bacteriophage has essentially genetically modified the bacterial cell, right? It's introduced genetic material into the bacterial cell genome that exists there stably. So it's not a plasmid in the kind of internal environment of the cell. It's actually integrated into the genome, so it's a part of the genome from this point onward. So once the bacterial cell is replicating and it's dividing, it is also replicating the bacteriophage genome, because the bacteriophage genome is within the bacterial genome. So this can go on and on and on and on and on. There can be a process called excision, so there is a process by which the bacteriophage genome can be cut out of the bacterial genome, but this doesn't always occur, and so a lot of the times this is a really stable integration, and for many rounds of cell replication it is maintained within the bacterial genome. Okay, so let's have another look at the comparisons here. So for the lytic virulent bacteriophage cycle, there is no prophage because this stage of integration that we just talked about doesn't occur. There is lysis because again, it's a lytic life cycle. This is what the virulent life cycle is. It's a similar concept to the replication of other lytic viruses, so viruses that infect eukaryotic cells and cause lysis in these cells, which is again something that we've said. So these viruses infect the cell, they produce copies of themselves, and in doing so they induce a huge amount of stress in that cell. So this cell all of a sudden has to produce large amounts of protein to meet the needs of this bacteriophage. So it's being forced to produce this protein. It's also being forced to replicate the bacteriophage genetic material. And again, there's a huge energy and resource cost to this, and this reduces the cell's ability to maintain its normal functions. And as a result, the cell fails to function and ultimately collapses, it lyses, it breaks apart. Bacteriophages also directly contribute to this process, so they encode within their genome some specific proteins, lysins and holins, that will mediate the lysis of the cell. So they will contribute to destabilizing the cell, which causes lysis, because again, this lysis of the cell is ultimately in the bacteriophage's interest, because it enables the release of huge numbers of bacteriophage particles that have been created in the cell. It enables their release into the environment where infection can continue in other cells. Okay, we also, as we've said, have the lysogenic life cycle that temperate bacteriophages can engage in. And again, this key central part of this is the formation of a prophage. So it's the integration of the bacteriophage genome into the bacterial genome. And this is really crucial from a disease perspective. So we need to remember this, because this is something that's going to be really important in the next part of the lecture when we talk about the role of these bacteriophages in human disease. Because remember, what we're talking about here is related to bacterial cells. So this is a process between bacteria and bacteriophages. But what we're going to look at in a few slides is how this process impacts humans, so how it impacts human health and disease. This idea of the prophage forming is essentially a type of horizontal gene transfer, so a process by which genes can be passed across a generation rather than up and down generations. Again, it's a natural process, however, it's something we can manipulate therapeutically and experimentally, and this is something else we'll look at as well. So what we can really think about here is this is a mechanism by which the bacterial cell is being genetically modified. That's essentially what's happening here, is genetic modification. Now, it's a natural form of genetic modification, but there's a lot of research interest in manipulating this experimentally, and that's something we'll think about later in the lecture. Okay, so those are the two life cycles that we need to understand. We need to understand this idea of virulent and temperate bacteriophages engaging in the lytic and lysogenic life cycles. Now what we want to do is tie this information to bacteriophages and disease. So we want to take what we've just learned and we want to understand it in the context of human health and disease. Because as we've said, those slides we were just looking at, they're just interactions between bacteriophages and bacteria. So why do we kind of care about this in the lecture about human health and disease and viruses? And that's what we're going to address now. Okay, so when we're thinking about bacteriophages and disease, we can think about this as really being a multi-layered question, isn't it? Because we can think about bacteriophages and disease from two perspectives. We can think about the disease caused in the bacteria that is infected, but also we want to think about the disease caused in human host, which is why we care about all this really. We're not particularly concerned with diseases in bacteria, are we? But if we are concerned with diseases in bacteria, we'd say that the primary way in which these bacteriophages can cause disease is the lysis, right? So it's the lytic life cycles that actually kills the bacteria. So obviously this is death for the bacterial cells, but we also want to think about the lysogenic life cycle, because this lysogenic life cycle is when there is a knock-on effect where the bacteriophages ultimately contribute to disease in humans. And the reason for this is, as we've said, bacteriophages essentially genetically modify the bacteria they infect if they are engaging in a lysogenic life cycle. So they are introducing genes into the bacteria, and these genes can contribute to the bacteria's ability to cause disease in a human host. So a bacteriophage can introduce pathogenic genes to bacteria, and in doing so they are essentially giving the bacteria the ability to cause disease in a human host. So there are bacterial species that are not capable of causing disease in a host normally. However, they acquire this capability when they are lysogenized by a bacteriophage. So when a bacteriophage infection occurs, so they can gain exotoxins, for example, so specific factors that directly enable them to induce disease, or they can sometimes gain antibiotic resistance. So again, this is something that enables them to successfully infect a host, so it contributes to their ability to cause disease because they're resistant to treatment. So this idea that bacteriophages can essentially genetically modify bacteria in such a way that they can contribute to their ability to cause disease is really important, and this is how we can tie bacteriophages to human disease, and this is the primary role that bacteriophages have in human disease. Again, we have the lytic life cycle on the right. So this is what's happening when bacteriophages are killing bacteria. Again, not particularly of interest to us. What is of interest to us is the lysogenic process. So again, the process by which bacteriophages genetically modify the bacteria cells they infect, which again involves integration, the formation of the prophage, and this prophage contains or can contain genes that confer either antibiotic resistance or some other ability to cause disease in a host. And when we look at this in detail and we look at different bacteria, we can see that there are a number of specific examples of bacteria that acquire the ability to cause disease after being lysogenized, so after being infected by bacteriophage, which introduces particular genes into the bacterial genome when it forms the prophage. So we'll work through some of those examples now. You'll see some important human diseases here. So this isn't really a niche thing, it's specific information, but it's really, really important because again, we're going to see some quite significant and impactful human diseases. Firstly, we have scarlet fever. So scarlet fever, sometimes called scarlatina, it is a bacterial infection. However, the bacteria that causes it is only able to cause the disease symptoms when it carries a specific gene that is introduced into the bacteria by a bacteriophage. So it's caused by the same bacteria that causes strep throat, so streptococcal pharyngitis, so pharyngitis, inflammation of the pharynx. Streptococcal, it's caused by Streptococcus species, specifically group A Streptococcus, and again it's the same bacteria that is causing this strep throat as is causing scarlet fever. It can be treated by antibiotics usually. So again, this is an important point. We're tying these diseases to bacteriophages, but we don't want to get confused and think that these are diseases that we can treat with antivirals just because the root cause is linked to a virus. The bacteriophage, so the virus, is not infecting the eukaryotic host, so antiviral treatment wouldn't make any sense, it wouldn't work. It's the bacteria that is infecting the host, and this bacteria itself has been lysogenized with bacteriophages. The therapeutic intervention that we want is to target the bacterial infection, so it's going to be antibiotics. Okay. So let's look at the pathogenesis in a little bit more detail. Again, we've said it's streptococcal, so it's caused by Streptococcus infection. And we've also said that a bacteriophage is responsible for causing the streptococcal species to acquire the ability to cause these disease symptoms. And again, to do this, the bacteriophage is introducing a gene into the bacterial genome. This gene encodes an exotoxin. There's a couple of different ones, but the most notable one is SPE8. It's a pyrogenic. So again, we can break that word up, pyrogenic, a pyrogenic exotoxin. And this is what causes or enables the bacteria to cause disease symptoms. And again, the bacteria is only carrying this gene when it has been lysogenized by a particular bacteriophage, in this case the T12 bacteriophage, which is a double-stranded DNA bacteriophage, which may be a member of the Cyphiviridae family. So again, we can look up this bacteriophage and read up a bit of information on it if we want. The main takeaway should be the key principle here, the idea that this bacteriophage is lysogenizing the bacteria, and in doing so is enabling the bacteria to produce an exotoxin that therefore enables the bacteria to cause disease in a host. Another clear example is Vibrio cholera, which causes cholera. So we talked about cholera in the past, so it's an example of a lower GI tract infection. You remember that central to the ability of Vibrio cholera to cause this infection is its ability to produce and release cholera toxin, which is an exotoxin that compromises the local environment in such a way that it induces massive amounts of diarrhea. It's transmitted by contaminated water, and we looked at a case study at some point where it was first found out that this was being that cholera was being transmitted by the contamination of drinking water with fecal waste. And again, what we can see here is the ability of Vibrio cholera to produce this crucial exotoxin is directly linked to whether or not the bacteria has been lysogenized by a bacteriophage. So in the case of cholera, the bacteriophage is CTX5 bacteriophage, and this particular bacteriophage carries the gene that encodes cholera toxin, sometimes referred to as CTX protein. It's exactly the same thing. It's the protein that functions as an exotoxin, and again compromises the local environment in the lower GI tract, causes this signaling cascade that results in water being drained and held into the lumen, so moving out from the cell's lining and not being able to move back into these cells lining the lower GI tract. And again, therefore, there's massive retention of water in the lumen, which causes watery diarrhea. The principles here are the same that we've already talked about, so it's just another example of the bacteria acquiring the ability to cause disease symptoms by the introduction of a specific gene into its genome by a bacteriophage. Okay, and next we want to think about diphtheria. So again, an infection that we've probably heard of before. Hopefully we're starting to see that when we're talking about bacteriophage in human disease, we're not talking about a niche area, we're not talking about a small number of diseases that maybe cause mild symptoms. We're talking about significant human diseases that have a real severe impact upon human health globally. So this is something we really do want to understand. And again, diphtheria is an example of one of these diseases. So it's caused by Corynebacterium diphtheria. So this is a bacteria. It typically infects the throat, and when an individual has an infection there's kind of general infection symptoms that can progress to a thick greyish dark coating at the back of the throat, which is essentially a compromisation of the local environment. It's dead cells and things like that caused directly by the infection. Now, it's an infection that's rare nowadays due to a really successful vaccination campaign in the last century, but it's still seen. It's definitely not eliminated, but as a direct consequence of really good levels of vaccination, levels of diphtheria cases have really significantly dropped. And again, the same principles when we're thinking about pathogenesis. The bacteria is able to cause disease when it is lysogenized by a particular phage. This particular phage introduces diphtheria toxin into the bacterial genome, and diphtheria toxin is able to inhibit bacterial protein synthesis. Next, we have dysentery. So dysentery is caused by Shigella dysentery. So this is bacteria. Sometimes you'll see the disease referred to shigellosis because it's caused by Shigella or bacillary dysentery. Diarrhea is the primary symptom. As we may know, dysentery is traditionally associated with diarrhea. It's spread in the same way as many other infections of the lower GI tract, particularly bacterial infections of the lower GI tract, so spread through the fecal oral route. And again, it's a bacterial infection, so treatment is centered on antibiotics. And again, the same principle is seen where the bacteria acquires the ability to cause this disease when it is lysogenized by a bacteriophage. So as we said, the bacteria is Shigella dysentery. The pathology is caused by the Shiga toxin. So this is the actual toxin that is introduced into the Shigella species. You'll sometimes see it referred to as STX, so Shiga toxin. STX phage is the bacteriophage that lysogenizes the bacteria. So this is the bacteriophage that introduces the gene into the bacteria. And the actual process that the toxin inhibits is protein synthesis. So Shiga toxin, when it's released by the bacteria, just disrupts protein synthesis because it interacts with ribosome subunits. And as we know, the ribosome is central to translation. So if the ribosome can't function, translation can't occur, and therefore protein synthesis is not able to take place. So again, just another example of the bacteria gaining the ability to cause disease due to this lysogenic life cycle of some bacteriophages. Next, we want to consider botulism. So botulism is a disease state caused by the bacterium Clostridium botulinum. This bacteria can release botulinum toxin. So this is a specific toxin that acts upon the nervous system. And when this toxin acts upon the nervous system, it induces a specific type of paralysis. It's essentially inhibiting the release of neurotransmitter. And in doing so, it's preventing signaling through the nervous system. And this is how it causes paralysis. It tends to be seen most often in infants. So we have this type of botulism called infant botulism. This is because infants don't have a fully formed microbial flora yet, so it's just easier for the bacteria, Clostridium botulinum, to colonize an infant if given the opportunity, because there's low levels of colonization resistance, which essentially means there's low levels of bacteria to compete with already there. So infant botulism is often how this is seen, and again the symptoms are related to the nervous system, so they're types of paralysis. Again, same principles are ringing true. So botulism is a disease state which is caused by the bacteria when the bacteria has been lysogenized by some specific bacteriophages. So botulism is a little bit more complicated than some of the other diseases we've looked at, because there's actually a range of different botulinum toxins, so a range of different toxins that can be implicated. And some of these are carried by bacteriophages, some aren't, but it's still a really useful and interesting example, because in a lot of cases it is the bacteriophage that introduces the gene for the particular botulinum toxin into the bacteria. So yeah, interesting for us to understand. And we have some of the specific toxins, C1 and D toxins, both botulinum toxins, that are introduced into the bacteria by bacteriophages, listed on the right of the slide here. Okay, so hopefully that was interesting, and hopefully now we're seeing that there is this central concept that ties bacteriophages to human disease, and it's the ability of the bacteriophages to introduce pathogenic genes into bacteria. Again, there's this little bit of confusion sometimes when we're thinking about antivirals and antibiotics, because we're talking about virology, we're talking about bacteriophages, but we're also saying the treatment is typically antibiotics. And again, the reason is the actual infection is a bacterial infection. It's just there is a viral role in that infection. And as we've said, that role is the introduction of pathogenic genes into the bacteria by the bacteriophage. So of course the ultimate treatment is going to be an antibacterial treatment, because the actual infection, despite the role of viruses, is a bacterial infection in a human host. Okay, so finally we want to think about the use of bacteriophages in research, because this is relevant to the role of bacteriophages in human health and disease, because it falls under bacteriophages in human health, because a significant amount of research into bacteriophages and into other microorganisms in general is focused on how can we improve human health, what information can we obtain that will help us to develop therapies and things like that. Okay, so as we've said, bacteriophages are essentially genetically modifying the bacteria they infect. So it's really useful for us to potentially think about ways we can manipulate this process. Okay, it's useful for us to think about bacteriophages as presenting a natural strategy for the delivery of genetic information into target cells. We start to see this when we're thinking about DNA vaccines, and potentially at some point RNA vaccines being delivered by this method as well. It's a method for us to deliver genetic material into target cells. Now obviously there's challenges there, because bacteriophages are only capable of infecting bacterial cells, but the idea is modifications can be made, and we can use bacteria to deliver genetic material into specific eukaryotic cells, and this genetic material, in the case of DNA and RNA vaccines, can encode specific antigens that the immune system then produces a reaction to, and this is the principle of vaccination. And the idea is that this type of vaccine delivered in this particular way would stimulate a really potent immune response. If we think more of the experimental research aspect, we can think about bacteriophages being used to deliver experimental genes into cells in culture. And again, we have the same issue of bacteriophages actually in vivo, in real life, targeting bacterial cells, but us wanting them to target eukaryotic cells because that's more useful for research. So there is a barrier to using this, but still the potential is there, and there are people really interested in this. in lab research, a lot of the time you want to introduce genetic material into cells because it's interesting to do that. If, for example, we have a protein that we're interested in and we don't really understand its function, one thing that we can do is we can overexpress that protein in a cell and see what happens to the cell. One way that we can force a cell to overexpress a protein is to just put lots and lots of DNA encoding that protein into the cell, and then this will presumably be expressed and the protein levels will increase. Again, the challenge here is introducing this genetic material into the cell, because it's really hard to do that by any strategy. Cells, membranes are really resistant to charged molecules passing through them. There's a good reason for this, because cells need to control their boundaries. They need to control their borders. They can't just have things moving in and out at random. It would be a disaster for the cell. And that's why the cell membrane functions, to keep certain things out and keep certain things in. But as a consequence of that, experimentally, it's really difficult to introduce genetic material to a cell. Bacteriophages potentially provide a strategy for us to present genetic information into a cell despite this barrier. Furthermore, as you should know from our discussion of the lysogenic life cycle, bacteriophages have the ability to introduce genetic material into cells in such a way that this genetic material is stably integrated into the cell genetic material, so into the cell genome. And again, this is really useful, because what we see experimentally is a lot of the time, even if we do manage to introduce genetic material into a cell experimentally, and we can do it, it's just a little bit complicated. You need some various reagents to compromise the cell membrane, but even if you do this, a lot of the time the genetic material is essentially pushed back out of the cell. It's almost spat back out because it doesn't belong there. And best case scenario, even if it stays in there, once the cell starts replicating, not all of the produced copies will have the new genetic information, because the new genetic information, it's not going to be copied like the host genome, because it's not part of the host cell genome. It's just kind of existing in the cell. So there's these barriers again. And one suggestion is, if we can get this integration event to occur, then that's one way to make sure the progeny of the target cell also carry the genetic information. So this is another reason why we're quite interested in bacteriophages. It will be really difficult to get that integration to happen in eukaryotic cells, but that integration example in bacterial cells will be useful experimentally, and that is something that can be done. When we want to integrate genetic material into a host cell that is eukaryotic, again, we have the challenges we talked about getting it into the cell that may be overcome by bacteriophages. But if we want the integration to occur, then rather than bacteriophages, because the host genome of eukaryotic cells is so different to that of bacterial cells, we lean towards lentiviral vectors. So these are vectors that are derived from retroviruses, because as we know, retroviruses integrate their genome into the host cell genome via the process of reverse transcription and integration. So a really different process to that which is conducted by bacteriophages, and it is a process that is specifically evolved to occur in eukaryotic cells. So really it's perfect, and that's something we do in the lab quite a lot. And we'll talk about that in later lectures in a lot of detail. Okay, great. Thanks very much everyone for listening, and I look forward to seeing everyone on Monday. Take care.